Hello, everyone. Last lecture, we focused on A/B toxins and saw how microbes can hijack specific cellular processes. Today, we're going to look at three additional mechanisms of damage. First, we'll examine toxins that directly destroy cell membranes. Second, we'll discuss superantigens, which overactivate the immune system. Third, we'll look at endotoxin, which triggers widespread inflammation. And by the end of the lecture, you should be able to um, describe how membrane disrupting toxins damage host cells, explain how superantigens and endotoxins trigger harmful immune responses, and connect toxin activity to disease symptom and outcome. Pardon me. All right. So here's our review slide. We've gone to this multiple times. We're now in this area of toxicity and tissue damage and disease. And just kind of as a refresher here, once a pathogen has successfully attached, invaded, and survived the immune response, uh, how does it actually cause disease? There are four major ways. We have our direct damage through toxins, which we started talking about last lecture, and we will continue here. Second, pathogens can cause indirect damage by triggering inflammation. Um, in this case, much of the injury actually comes from our own host immune response. The immune system is trying to eliminate the infection, but the inflammatory response can also damage healthy tissues. Third, pathogens can create a nutrient and metabolic burden, which we're not really going to focus on in these lectures, but microbes require nutrients to grow and reproduce, and they often compete with host cells for resources such as iron and other essential nutrients. You can really think of infection as being a nutrient acquisition strategy, whereas we may hunt and perform agriculture, or a grasshopper is going to eat grass. Um, Pathogenic microbes are trying to obtain their nutrients in a different way, and that way is by getting them from our bodies or whatever their host is. So finally, some pathogens can cause damage through physical invasion or cytolysis. As they spread through tissues, enter cells, or replicate inside cells, they can directly destroy host structures. So in this lecture, we're going to focus primarily on toxin damage and inflammation. Let's talk a little bit about membrane disrupting toxins. Unlike AB toxins, which alter cellular function, membrane disrupting toxins simply destroy cells. One strategy is to punch holes in membranes. These are called pore-forming toxins. A classic example is streptolysin O, produced by Streptococcus pyogenes. Um, Multiple toxin molecules assemble together and create large pores in the membrane. Once those holes form, ions and cellular contents leak out and the cell dies. Another strategy is to chemically digest the membrane itself. Alpha toxin from Clostridium perfringens is a phospholipase. Rather than creating holes, it breaks apart phospholipids that make up the membrane. The membrane falls apart because they're no longer built together, and then the cell rapidly lyses. These are different mechanisms that have the same outcome, which is dead cells and damaged tissues, which is in complete opposite of those AB toxins that were primarily changing cellular function. Most antigens activate only a tiny fraction of T cells. However, superantigens bypass this normal specificity. They physically connect to MHC2 molecules on antigen-presenting cells with T cell receptors, regardless of what antigen is being presented. As a result, 20 to 30% of all T cells can become activated simultaneously. This creates a massive cytokine release known as a cytokine storm. Importantly, the toxin itself is not directly destroying tissue. Instead, the immune system becomes overactivated and begins causing widespread inflammation throughout the body. This can lead to fever, rash, capillary leakage, hypotension, and potentially shock. Endotoxin works very differently from exotoxins. Endotoxin is not a secreted protein. Instead, it's part of the outer membrane of gram-negative bacteria. The biologically active portion is lipid A, which is found in lipopolysaccharide, also known as LPS. When gram-negative bacteria grow, shed membrane fragments, or die, lipid A is released. Innate immune cells recognize lipid A as a danger signal through receptors such as TLR4. This triggers productions of inflammatory cytokines. So, unlike AB toxins or membrane toxins, endotoxin does not have a specific cellular target. Its effects come from activating inflammation throughout the body. At low levels, this can be beneficial. At high levels, it can cause fever, vasodilation, hypotension, um, disseminated intravascular coagulation, and even septic shock. One useful way to think about toxins is whether they act locally or systemically. Membrane disrupting toxins usually act locally. The bacteria produce the toxin near a tissue, cells die, and localized damage occurs. Superantigens and endotoxin are different. Their effects are systemic because they trigger immune responses that spread throughout the body. This distinction helps explain why some infections produce localized tissue destruction while others produce fever, shock, and multi organ dysfunction. So think about something like a wound infection. Like you might have irritation around that wound, but you probably didn't get a fever, right? But when you have a like a cold or like flu, which are not bacterial diseases, but they are more systemic as well, you get this fever response, which is that inflammation that's coming from the immune system. An important concept in microbiology is that not all damage comes directly from the pathogen. Neutrophils and macrophages release reactive oxygen species, proteases, and inflammatory mediators to destroy microbes. These defenses are essential for survival. However, they are not perfectly selective. Nearby host tissues can be injured as collateral damage. In many severe infections, some of the symptoms we observe result not only from microbial production, but also from the host response itself. So, today we examine three major mechanisms of toxin mediated disease. Membrane disrupting toxins destroy cells directly by damaging their membranes. Superantigens overactivate the immune system and trigger cytokine storms. Endotoxin activates innate immunity and causes widespread inflammation. The major theme is that pathogens can cause disease either by directly damaging tissues or provoking host response that becomes harmful. Many of the most severe symptoms of infection, including fever, shock, and organ failure, are not simply caused by the pathogen itself, but by the interaction between microbial virulence factors and the host immune response. With that, I thank you for attending this lecture, and I think this is the last one of the semester, so maybe I'll see you in another class. Bye bye.